Today's class will be about how to build a biological oscillator. So we're going to switch gears. But before that, let's all take a nice deep sigh of relief. We're here. Okay, we already did two things. Another nice sigh of relief. Welcome. You know, up to now, we've discussed quite a bit about biological circuits that try to keep a balance. We talked last time about how to keep blood glucose balanced, insulin, dynamic compensation, how beta cells can expand to compensate for varying parameters. Remember that? We talked about integral feedback for exact adaptation. The circuits we talked about so far had maybe one pulse like that, reached steady state. Today, we're going to talk about the wild world of biological oscillations. The thing will go up, down, then back up, down, down, up, like that. They're going to be, still there's some order in oscillation, right? We want a frequency, certain frequency, maybe we want to control it, maybe we want to tune it, like our heart beating. If our heart's beating, it's nice to have 60 beats per minute, but maybe if we're excited or exercising, we want it to be 180, 3 times faster, but we still want the heart to beat, right? So we want to tune the frequency. How to build, let's take a nice deep sigh of relief. Perfect timing. So I just want to start off, because music is also an oscillation, a song, and I want to ask you to be backup singers this time. Okay, so your text will be roll, 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 roll. This song by The Doors, right? And it'll be like this. I'll say roll, roll, baby, roll. You go roll, roll. Okay, let's just practice it. Roll, roll. Can you say that? Shalosh, arba, ve. Roll, roll. Okay, good rhythm. I said roll, baby, roll. Okay, then you go roll, roll. It's not easy, but this is an advanced class, right? I said roll, baby, roll, roll, roll. I said roll, baby, roll. Roll, roll. I said roll, baby, roll. Roll, roll. I said roll, yeah. Oh, all night long. That's what oscillators do, okay? So we'll get back to that at the end of the class. So remember your part. You know, what kind of biological oscillators do we know? So I talked about the heart beating. We have inside us an amazing clock called circadian rhythm that keeps track of the time of day. Nice deep sigh of relief. It's the time of day, Tuesday at 11:15, where we start the class. So our body knows, to give us a pulse of cortisol, right? In the morning when we wake up, it has a circadian clock in our brain. It's an internal model of the 24-hour clock. Very important for organisms to have an external model of the varying world. We have cell cycle oscillators. So if we have a single cell, at a certain point, it divides its chromosomes, turns into two cells, and vice versa. So like when an embryo divides into all the cells to make a baby, there's billions of divisions like that with very precise time scale. There are neurons trains, spike trains. So if you record a neuron, it sounds like that. There's spikes of electrical activity with well-defined timing. And it's a tunable oscillator. It could be sometimes sometimes like that. So tuning the frequency. And when you look at the heartbeats, also you have this stereotype form with tunable frequency. And there's calcium oscillators, glycolytic oscillators, and many kinds of oscillators in the cell. More and more are being discovered. And what I want to talk about, there's different circuits to do it. I want to talk about what kind of biological circuits can give you oscillations, and what, among all the possibilities, what circuits by evolution seems to choose again and again, and why. So these will be our principles of how to build a biological oscillator. That's our plan. So by the end of this class, you'll be able to look at a biological oscillator and maybe understand a little how it must be working, why there are the arrows there in the circuit diagram, and have some concepts about oscillations. Now I want to, so this was review oscillator. So we begin by noticing that at the heart of every oscillator, there has to be a negative feedback loop. So there has to be something that takes us, if it's a protein, when the protein is high concentration, something that brings it down. And then when it's at low concentration, something that brings it back up. that's a negative feedback loop, right? When it's high, let's say it shuts off its own production. Then when it's low, it's made again. But when it's high, it shuts off its own production. So inside every biological oscillator, there's a negative feedback loop. But just having negative feedback is not enough, okay? And I just want to demonstrate that with an analogy or a metaphor. Metaphor is very important in science. The metaphor is an experience I had when I was a child in my shower in Kfar Saba, where I was growing up. I think I told you about this shower before. So this shower, the negative feedback loop is I walk in, I turn on the water, and it's cold. Okay? So the negative feedback is me turning the water to the hot. Now, today's shower, where I live now on Weizmann, is really the modern shower. So I just turn it back, and I find the sweet spot, and there's no oscillations. But in the old shower, okay, it took a while for the water to understand what I want, right? So I would go to hot, and so it's not getting hot. So I go hot, hot, and then it's boiling, and then I turn it back to cold, and then it's freezing, and turn it back to hot, and it's boiling like that. So that was the delay. So negative feedback alone, if it's very fast, won't give you oscillations. But if you add in a delay, it will. Okay, so let's work with this little example. Any questions about that?
In my shower in Haifa, it was also stochastic. So.
Why was it stochastic? Yeah, so you say it was stochastic. So that's noise, right? We'll talk about that. So delays are good for oscillation. We'll see also that noise is good for oscillation. Good. So I want to start off our discussion with negative feedback. Is not enough. For example, the simplest negative feedback, protein X inhibiting its own production like that. So we remembered, we talked about this in one of the first lectures, that this has a tendency to go to X steady state. So if you start somewhere, it goes to steady X steady state. In fact, it has a fast response time. Do you remember? It speeds responses, very stable to noise, and there's no oscillation here. This is a return. And if I start high, it'll also go back. There's no oscillations. And in general, I just want to say that any equation that has only one variable, so this is any dynamic equation, this is the rate of change of x, cannot have an oscillation. And I just want to remind you the way and I'll show you why. So in the negative autoregulation, we had removal, and we had some kind of a function where x represses its own expression. So this is x repressing its own expression and degrading. So we had the degradation rate. This is all stuff I hope you remember from, I think, the second lecture. And we had these two rates. So this is the production. This is removal. And we had this point where production equals removal, where the two lines crossed. That's balance, right? Production equals removal. That's the steady state. And we notice that if we're below the steady state here, production is more than removal, so we move, x moves up. And if we're here in this region, removal is more than production, so there's less of x. And that's it. You converge. Now, why do I say that you can never have oscillations, no matter what the shape of these lines are? The reason is that no matter what the shape of the lines are, any point on the x-axis has an arrow, either moving this way or that way in a one-dimensional system. Now, for an oscillation, that means that if you're here, you know that you should move either that way or that way. If this equation has no time anywhere inside, no time-varying components, in order to have an oscillation, you need to go, at this point, to move that way some of the time, and then that way some other time. Otherwise, it's not an oscillation. But that means you have to have two arrows in one point. And that's impossible, because the arrow direction is determined by the rate. It's either there or there in one dimension. You can't have an oscillation in a one-dimensional system that doesn't have time-varying parameters. Of course, you can add some time-varying parameters or delays, but that's no longer a one-dimensional system. Did I explain myself? So, yeah.
But this is because you assume the function has to be...
Yeah, I'm talking here about any. Now, if I replace it with any function that I want, any function.
Just like this. Just very,
Oh, good. Okay, let's challenge.
very low, no mid.
Okay, very high.
Would you overshoot?
Very. Come here. Draw it. Do you want to draw it? Because I can. Yeah.
Way up and starts way down.
The removal production, way up, starts
The
up.
difference between them, I don't.
Oh, the difference
How
between
you-
them.
How you
Okay.
sort of straight and goes way up when you're sort of close to this. No, yeah, no.
Okay, let's give her some applause. We have to challenge every idea, right? Your name is Sigal. And Sigal, what's your last name?
Pellet Leviathan.
And where? And where do you work?
Eran Segal.
Ilan Segal's lab. Great.
If you had something like that,
Yeah.
sort of, that's,
Yeah.
so basically you would overshoot every time. And if you overshoot, then you go back and forth.
Great.
Because from here, you wouldn't stop here. Because
Yeah.
when you get close, you really want to
Yeah.
go over.
Yeah, I got it.
Etc.
Yeah. So you're saying, if you get here, you want to move here so fast
That
that you overshoot.
the point.
Yeah.
Yeah.
So if there's any delays,
I'm not sure
yeah.
it's biologically possible.
Yeah,
but
okay,
it's
great. Now,
theoretically
this is really an extreme situation. And I want to say that in real life, there will always be a delay that will do that. But in the strict mathematical sense, each point here, this is like a singularity, right? You move this way here, and you move that way there.
Right.
And this point is kind of a stable point.
Well, not if it goes to infinity and minus infinity, and then it's not a stable point.
Yeah.
You overshoot by
Yeah,
definition.
so this, I think, is like an...
I don't
is
think
it-
it's biologically
yeah,
possible.
yeah.
But
But still, I would say that if you strictly stick to one-dimensional equations, you still have to decide at each point whether you're moving this way or that way without any delays. I mean, instantaneous. And then you can't at the same point sometimes move this way and
no,
sometimes
but
move
there
that
is
way.
no point here where you are. I mean, this point does not exist. I mean, it's a sort of a...
Yeah, there's another problem
It's a
here.
point
That
you can't be-
this is like not a continuous
Yeah, that's
function.
what I'm saying.
Yeah.
You can only
Yeah.
do this with a non-continuous function. You really have to go to infinity and minus infinity in order to overshoot.
Thank you. Okay, so I have to think about that. I was thinking mostly of continuous functions, but thank you. מחיאות כפיים. All right, so we need to have a delay here, basically. And if we have a delay, indeed, negative feedback with a single node can oscillate. The typical case you add a delay in biology is by making a two-node feedback loop. So you have X that makes something, and that something degrades X. That's a two-node negative feedback loop. And we discussed these two a little bit. And if I have this circuit here, we have a delay. It's like in the shower. And now we solve these equations, and we discover the following situation. And now it depends a lot on the parameters. So I'm going to put some parameters on these arrows. I'll call this beta 1 and beta 2. These will be the feedback strengths. And then we have the removal rates, alpha 1 and alpha 2. So we're thinking something like dx dt equals f of y minus alpha 1x, and dy dt equals g of x minus alpha 2 y, where f of y is a decreasing function and and g of x is an increasing function because there's a activation and inhibition arrow. And these are the removal rates. Equations here don't matter too much. What you can get in this situation, this is time, is that x does this. And you can say, wow, great, we have an oscillation. It's starting to oscillate. Great, great, great. But then it stops. So that's called a damped oscillation. And y is kind of oscillating in counter phase here because when y is high, x is low, etc. Or for other parameters, you can have just like the one node case, this is called overdamped. There's not a hint of going above and below. So these are the only options you can have for this circuit. You can have damped, overdamped, or damped. The amplitude here goes down exponentially to 0, so the oscillations kind of damp out. And people who study physics know this from a kind of linear oscillator with dissipation. I want to say in physics, we study a lot of times linear oscillations that have none of these removal terms, And then they can oscillate. But once you add a little bit of removal terms, that's like dissipation, you always get damped oscillations. So this is, in physics, is a little different. Linear oscillators, because of this conservation of energy, are often studied. But in biology, you always have some removal term. So so we still have no oscillations. It's a little bit frustrating, right? Even when we add another node like that. And what, I just want to say, but this is a hint, so we're going to build on this. We want to amplify those oscillations a little bit. We want to keep them sustained. But what's the difference between the parameters for this and this, the parameters? So there's a condition. You can actually solve analytically the linear stability equations here, which I decided not to do in the class because it's, well, maybe we'll do it in the exercise. And you can see that in order to get damped oscillations, you need that alpha 1 minus alpha 2 squared be smaller than 4 beta 1 beta 2. So there's a condition for the parameters. Now, what's this alpha 1 minus alpha 2 squared? This is the difference in removal rates. And this is the feedback strength. So in order to get oscillations, and this is going to make a lot of sense, we need the two timescales of x and y to be similar. If alpha 1 and alpha 2 are 0, minus alpha 2 is 0, alpha 1 equals alpha 2, you always get damped oscillations in this circuit. Why? Why is that? Because if one of these reactions is much faster, if one of these x's responds much faster than the other, we can basically forget about it, and we have a single node negative feedback. It's like if y is, one x changes, y immediately responds, it's like x is controlling itself. And then we get back to the perfect shower situation with no delay. Only if the two timescales are the same, we can be justified in saying, okay, there's two nodes here because not one much faster than the other. And then you get oscillations. And then on the other hand, here is the feedback strength. The bigger these arrows are, this is the more. So of course, if we had no negative feedback, x and y would just reach a steady state and won't affect each other. When beta 1 is 0 or beta 2 is 0, we don't have one of these arrows, and we have, let's say, a circuit like this, which of course can't oscillate if this beta 2 equals 0. So that's why this product is nice. If one of these is 0, no way to get damped oscillations. But if they're both fine, then we can get, the feedback is strong, and the timescales are matched. We have more oscillatory situations. So that's, I think, that's a general principle that carries throughout oscillators, is that the timescales need to, of the different, or in these kind of oscillators, the timescales need to be matched, and the feedback needs to be strong. And I just want to say one more thing about these betas. These betas are related to these functions. They're, in fact, there's a fixed point, and it's related to the slope of these fixed points. It goes like df dy at y steady state. And beta 2 goes like dg dx at x steady state. And the more steep, the steeper these regulation functions, the bigger the beta, and the more oscillatory the system. And I just want to go back to the shower. What do I mean a steep regulation function? In my old shower, not only was there delays, but the knob that controlled the temperature was also kind of an all or none thing. So there was a small range where you actually, so it was cold, 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 medium hot, 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 hot. And that made it more difficult to hit the right point. So that steepness is also plays a role in these oscillations. So what are any questions here? Yeah. This one? The relation between beta and the slopes? What are these squiggles here? Yeah, so this is the df dy. So this is the slope of f and g. The slope means, so when I wrote down my regulation function, so let me see. Okay, let's erase here and give you a little bit more detail. זמן לא יחכה לאיש. But we have time in this class. When we have x activates y and y inhibits x, then here there's a function g of x, and here is a function f of y. So g of x is the rate that x activates y. And f of y is a decreasing function because it's the rate that y inhibits x. And the important thing is the slope of these functions. The slope of these functions. Where? Around the steady state point. And the reason is that when you do a linear analysis, the slope determines, if you make a small perturbation, the slope is how quickly it'll return to steady state. And there's a way to just take down the system of equations that I wrote down here, linearize it, get a matrix, and then the eigenvalues of the matrix determine the stability. And I decided not to do that in the class, unless you guys really want to. I don't think you do? Who wants it to see it? Linear stability analysis. Who knows linear stability analysis from physics or somewhere? They want to see you? So like half the class, more or less. It's cool because you can get conditions like this about when it's damped or oscillating or overdamped, and you can understand some things. I just want to draw another picture. This is, so remember, in a one-dimensional system, we just had x, and we had, let's say, a fixed point, and you had an arrow at each point, and it can only move one way. So in 1D system, no oscillation, because the arrows can only move one way at each given point. But in 2 dimensions, you can have oscillations. You can easily have things like that. But in our system, they don't quite make it, and they spiral around and down into the fixed point. That's what damped oscillations look like. This is called a spiral fixed point. And the overdamped oscillation case, you have a fixed point, and just wherever you are, you go to it, like that. This is called a stable fixed point. So that's the options for this equation, a spiral fixed point or a stable fixed point, and that's what it can do. Now, I just want to tell you that in around 2003 or so, we decided to look at a negative feedback loop and measure these damped oscillations. And because at that time there was the ability to connect fluorescent proteins to actual proteins in the cell and measure in single cells the protein dynamics. That was around, turn of the century that became possible to do. And we chose a very important feedback loop that for in the cell that has to do with cancer. P53 is this gene that responds to cell damage, and it activates genes telling the cell to stop dividing, repair the damage, and if there's too much damage, to kill itself. So it's a very important gene to keep the cell from basically dividing when its DNA is damaged, because then you get mutations, and then you can get cancer, etc. And what happens in cancer is that 50% of all cancers have a mutation in P53. So it's the gene that's most mutated in cancer, making the cell not able to detect its own damage, you can say, and kill itself. So that's why it was so important, like very highly studied. And there were labs, Moshe Oren and Devar Rotter in our department that studied it. And we were system biologists, and Galit Lav, who was a postdoc, decided to do the following, to connect this one to a green fluorescent protein. It's this protein connect to a green fluorescent, and this protein connect to a red fluorescent protein. And now in the same cell, you can see green and red colors under microscope. And she gave a pulse of damage that made DNA damage, and she photographed the cells. And in fact, then we didn't have autofocus, so she was quite heroic. She had like put a bed in the lab, and every 15 minutes she got up to focus the microscope. And 1 year later, we had an autofocus microscope. You don't need to do that, but because we wanted to see these damped oscillations, and we were building, engineers know how to make this optimal ratio between the frequency and the decay in order to get like, there's some principles about damped oscillations we wanted to see, and we got a real surprise. Of course, it's just a two-node negative feedback loop, and so we want to show you the movie. Maybe we can drop the screen. You know, Galit Lahav was just announced. She's now head of the Department of Systems Biology at Harvard Medical School. She's an amazing researcher. And also she's a very nurturing kind of scientist. So she spreads a lot of, let's say, attention to human beings around her. So I'm expecting great things from you, Galit, if you're watching this. Roll, baby, roll. So, do you think we can play it? Maybe we can, should we turn off the light also? Oh, I should play it, just play it, right? Huh? חכות רגע? רואים משהו? Maybe any questions so far before we play the movie? Yeah.
The condition with alpha 1, alpha 2, and beta, where does it come from?
This comes from taking these equations, linearizing them around the fixed point, and asking when do the eigenvalues have imaginary part. When the eigenvalues have imaginary part, e to the lambda t goes into sine, cosine plus I. And that point where they become imaginary is the point where the stable fixed point turns into a spiral fixed point. And that has to do with, it's basically the trace of the Jacobian matrix and the determinant of the Jacobian matrix have to obey this condition. Did I answer your question? Yeah. So I know it's like sounds. Okay, so let's. So okay, so here's Galit. She was taking a movie of the cells, staying up all night to do the. Is it starting at the beginning? No, it's not. Okay. Okay, so I'll play it a few times. So what you see, you don't see anything. What you're going to see is a black square here with one cell in it. The cell you'll see green and red are P53 and Mdm2, and P53 goes to the nucleus. So you see like a green spot. P53 and Mdm2. And on the right hand, you see a graph that's plotting the green and red fluorescence intensity as a function of time. We're experiencing a technical difficulty. Yeah.
What does MDM2 do?
Ah, okay. Thanks. So maybe I'll give more details. MDM2 is basically a negative regulator of P53. It binds to P53, causes it to be ubiquitinated and degraded. So this is degraded. And this is transcription. So P53 is a transcription factor. It binds to the promoter of MDM2 and causes it to be produced. So MDM2 is produced, and then it targets P53 for degradation. That's the way this works. And P53 is a transcription factor that regulates hundreds of genes that have to do with DNA repair, stopping the cell cycle, killing the cell killing itself, cell becoming senescent, which means kind of stops dividing. And oh, it's embarrassing. Not too bad. What do you think?
You can call somebody.
Yeah. Yeah, we'll just continue. All right. Sorry. Maybe you can try to figure it out. Is it working? No, just play. Huh? Yeah, I can send you the link. And you want to look at it on your iPhone? Yeah? Yeah, great idea. Why not? This is, after all, 2018, right? We're not in 2000. Okay, check it out. Take your iPhone out. Yeah, let's do that too. So this is everything together. So go to, let's see, try to write to go to Uri Alon webpage. Just type Uri Alon, and then you get to my my home page. Yeah. Okay. When you get to my home page, there's it looks like this, Uri Alon Lab. Then on the top there's a movies. Movies. Click on movies. When you get to movies, you see, by the way, the courses, the videos for this course from 2011, 2014, and 2018. And then you can go to to dynamic proteomics. There's a menu that says dynamic proteomics. Proteomics means study of proteins. And then you see a gallery of movies. And kind of in the middle, there's a P53 Mdm2 feedback. Am I going too fast? So click on that and play it. Yeah, I'll show you.
No, I got it.
You got it.
Just takes time.
Okay. All right. So I am. Can anybody describe what you see there? You see oscillations. What else do you see? Is it kind of opposite phase? Great. Is it very stable oscillations, very precise, or more noisy? More noisy. Are the peaks rounded or sharp? So and so. Okay, so we, to our surprise, we saw oscillations. So we saw P53 and Mdm2 go up, down, up, down, up, down, and the Mdm2 does the opposite. And the time scale is about 6 hours. Okay, so you do the DNA damage, Mdm2 goes into the nucleus and out. That takes 6 hours. P53 P53 goes into the nucleus, makes Mdm2. Mdm2 causes P53 to be degraded. Then Mdm2 is not made anymore. It's just degraded. Then P53 goes up. So that was surprising for us. And we asked, and there's no known, I mean, there's, of course, part of a huge circuit, but this wasn't like obvious biology beyond this that could make us think why it oscillates. And then we got it. We got it. That in fact what's going on here is a spiral fixed point like that. But in all this discussion, we didn't add noise so far. So noise is, as I told you, when proteins are made, always there's randomness in this process. And when you add noise to a spiral fixed point or damped oscillation, a damped oscillation, you get noise-induced oscillations. And let me just explain that. X and Y spiral down, right? But if there's noise, it kind of kicks it. Noise always kicks you in some direction, some random direction, and keeps you from crashing into the planet here. Okay? Noise kicks you, and you just spin around like that, and you don't crash down. Noise-induced oscillations, so it turns this into this. So in other words, noise-induced oscillations, if you take this damped oscillator and push it away from steady state, it'll do that. But noise always pushes it away from steady state. So it's always trying to damp back down. But then noise pushes it some more. So these noise-induced oscillations have the following properties. They have a frequency that's quite precise, actually. And it's equal to the frequency of the spiral fixed point, which has to do with the imaginary part of the eigenvalues I talked about. The imaginary part of the eigenvalues. And so if this has a typical frequency, that's the frequency you'll see. But the amplitude is noisy because it goes like the noise amplitude. So by the way, I just want to make sure we are on the same page. Frequency means the typical time So if I talk about the typical time between two minima, like that, that's called the period, t, and the frequency is 1 over t. It's 2 pi over t usually. So it's like how many oscillations it makes in a unit time. And amplitude is the height of the pulses. So frequency and amplitude, yeah.
So I might be pushing a little bit ahead, but you mentioned that this is, generally speaking, a noisy process.
Yeah.
But from what I know about dynamics, this all depends on the noise. I mean, is this white noise? Is this concentrated around a certain frequency? What does the noise look like?
So the question is, what does the noise look like? So we don't really know enough so far about the spectrum of noises in the cell. But I do want to say that once you have a damped oscillator like that with a typical frequency, it picks up that frequency from the noise. So even if you have white noise, it amplifies that frequency. It's like a resonator. And that's what we think happens. And so it doesn't matter so much what the noise frequency is, mostly how much noise there is at that 6-hour time band. Yeah.
So the cell is using, the system uses the noise?
Yes. Here noise is useful, you can say. We still didn't talk about why it might be useful for P53 and Mdm2 to oscillate. It's an interesting question, but if you want to build an oscillator, one way to do it is to have a damped oscillator plus noise. And once we saw that, this possibility seems to apply especially for the situation like this of transcription factors in cells. In fact, now it's become a very diverse field. There was P53 and another transcription factor, NF-kappa B, which has to do with immune responses around 2002, 3, 4 were discovered to oscillate. And since then, there's many. So I would say that almost typically, transcription factors inside human cells will, if you have a family of transcription factors, at least one of them will oscillate. That's in the immune system and in development, and it's so this. Our eyes were open to that possibility because of the ability to look at individual cells. Because before that, you took millions of cells and made juice out of them, and you measured one time point like that. And if the oscillations are a little bit not synchronized, you just can't see it. So in gels, or like when you average many cells, and you put some DNA damage, you saw damped oscillations if you average many cells. Because the individual cells aren't exactly periodic and they basically cancel out. And that was the state of the art around 2000. We did that experiment. The first paper we did on P53 and Mdm2, we did these gels and we saw damped oscillation. That's why we thought, let's look at the damped oscillation in single cells. But then we saw that they actually oscillate. So there's nowadays, with single cell transcription, et cetera, there's technology to look at individual cells. It's just changing the way we think about almost every part of biology. Why should transcription factors oscillate like that is also a very interesting question. And there's several theories. And I just want to say that the typical situation is that you have, let's say for P53 and Mdm2, if you give gamma radiation that makes double-stranded breaks, you get oscillations. But if you give UV radiation that makes other kind of DNA damage, you get a graded situation. The more UV you give, the more P53 is made. The more gamma you give, the more fraction of cells oscillate. So it's a different encoding of signal. One kind of encoding is what fraction of cell oscillates. Another kind of encoding with another kind of damage is just regular how much P53 is made. So it's like, yeah.
I don't see why when you average many cells, you see oscillations at all. Wouldn't they cancel out within the cells that are high and some?
So the question is, when you average many cells, and they're not synchronized at all, you would just see a flat line. Here in this experiment, you give gamma radiation time t equals 0. So all cells do the first pulse together, then the second pulse a little bit apart, and then, yeah. So if you do something to make the cells start oscillate, you see damped oscillation on average, and in each individual cell you see noisy, full oscillations like that. Okay. There's Yeah.
When you said about the data regulation, you said that if you give gamma radiation, then only part of the cells,
Yeah.
only
Start.
cellular cells
Oscillate. So at low doses of gamma, it looks like the cells are either have no double-stranded breaks or can repair them. At high doses, and so those that can't oscillate, and cells that we give a lot of gamma, basically all the cells oscillate. So the fraction of cells increases that oscillate increases with the gamma dose. It's like the photoelectric. It's a kind of an all or none or discrete kind of sensing. This is a different ball game than what we talked about so far, where you have these continuous measurements. So the world of single cells has these surprises, and I can talk about that for a long time because this is an active area of research. So sometimes you get pulses, like crazy one is another example, where they look like this, pulses like this, and the number of pulses goes as the signal. And so the signal is encoded here, not by refraction of cells, but how many pulses you get. And neurons is like that too. Sometimes you have. So that's another kind of transcription factor that goes into the nucleus. This is in yeast that oscillates like that. And the downstream genes of this crazy one have a big benefit from this because gene expression is not by the amount, but just by the fraction of time that it's high. So this is like a kind of coding where each spike gives a little bit of gene expression, and you can control that way gene expression, not by amplitude, which could be noisy, but by timing, which could be quite accurate. So this is called frequency modulation, FM. And as opposed to a situation where we have amplitude modulation. The signal modulates the amplitude. Here the signal modulates the frequency. So at low signal you have something like this. At high signal you have something like this. Low, high. Here at low signal you have something like this. High signal you have something like this. And they have advantages and disadvantages. FM is disadvantageous at low signal because then pulses are very rare. And you either have a pulse or you don't, and that could be quite noisy. But high signal is very accurate. So at low signal, one might use AM and switch to FM at high signal, and indeed that's what happens in some systems where you have two isoforms, one of them oscillates, the other one doesn't. One has FM, the other one has AM, like one was discovered by Nir Friedman here at Weizmann, NFAT 1 and NFAT 4, two transcription factors in the immune system. They regulate genes. This one as FM and this one as AM to the same signals, so that you can get the best of both worlds by combining both of them. And this is like an emerging field of digital thinking, you can say, inside the cells, based on these oscillators. It's just like in electronics, I guess. There's clocks, right? Accurate oscillators. You use digital signals sometimes because you can get a good handle on the computation. Biological oscillations are more noisy. It's not like in electronics, but it opens the possibility that we have analog and digital computation together in the cell. Isn't that amazing? I don't know. Yeah.
When you said there is noise, so noise exists all the time, would it spontaneously start to oscillate? Or what will make it end? What will make it like stop oscillating?
Oh, yeah.
Noise that takes you from its spiral.
Yeah, good. So, if there's like a signal, like saying DNA damage, and you need that signal for P53 to be active as a transcription factor, then it'll only oscillate when there's DNA damage around. So things like that. So in fact, that's what happens for P53. There's like these upstream signals that sense DNA damage and modify P53. And there's different sensors for different kind of damage.
So what did you say was the advantage of FM?
FM advantage. Suppose you have two genes that are regulated. This is Michael Elowitz, called it bang bang control. So these spikes are either on or off. So these genes, this is a X, our transcription factor. So if these genes have some input function, so this is how X controls this gene. In a normal situation, you have a signal and it sets X at some intermediate level like that. So this is, let's say, gene 1, and this is gene 2. And as the signal changes, you get some gene 1 and gene 2. And you need to really precisely tune these functions in order to get the good ratios between them at every signal. Continuous, okay, AM requires good tuning of every f of x for every gene. But bang bang, this pulse situation, means that you're either here or here. So all the genes are either turned on or off. And now by just setting the number of pulses like that, you can set the ratios between these genes very precisely. All you need to care about is their production at maximum signal, you can say. Times the fraction of time the pulse is there. So FM gives good ratios between genes, which is this level here. Let's call it max 1 and max 2. Max 1 times number of pulses divided by max 2 times number of pulses. And you just get that the ratios are not dependent on all the shape of this function, which could be quite elaborate. So this is a kind of an idea about why bang-bang control works. And that's also in electronics. In electronics, when you want to control a device, digital electronics means you either have 0 volts or a lot of volts, and you can get rid of all the details in the middle. You become robust to all the shapes, the transitions, etc. It's either on or off, and you can get better control. I don't know. It's as I said, an open field of research. Why things oscillate with transcription factor? It just came as a surprise. We're still trying to digest it. Okay, and I want to finish up by telling you about the way that most biological oscillators are built. Because they have another principle that's in addition to everything I said. So if we summarize what I said so far. I said that you need a negative feedback loop. Delays help. Equal time scale between the two arms helps. Steepness helps. And noise helps. Do you agree? Most biological oscillators, like in the heart, in the circadian clock, in the cell cycle, neurons have a motif that is this motif. It's a negative feedback loop coupled to a positive autoregulation. So positive plus negative. And the kind of oscillations you get for them are different from our oscillations we looked so far, these delay oscillators we looked at so far. And let's say, I forgot to tell you that if you add a 3 loop like that, let's say a negative feedback loop made of 3 repressors, that's called the repressilator, then you don't even need noise, you get oscillations. You get oscillations that look like this, typically. They're nice, rounded oscillations. They can be noisy sometimes, even though So Michael Elwitz built this by hooking up 3 repressors in E. coli and hooking up to green fluorescent protein, and the bacteria turned black, green, like that with 8-hour period. Even though the bacteria divided in between, they carried the phase to their children. And recently a new version was built by Johan Paulsen that oscillates much more precisely. You can even see it in a test tube because they're so synchronized. But this motif here gives you typically different types of behavior. It looks like this. X rises gradually, then it explodes, and then it falls. And rises, explodes, falls. Explodes, falls. And Y rises here, makes X go down, and then falls itself. It rises here. So in this oscillator, X and Y are kind of together. In this oscillator, X and Y usually out of phase. Here they're together. And it's spiky. X and Y are together. And I want to say, in this kind of oscillator, it's hard to change frequency without losing amplitude. So if I start playing with, because the frequency of this is basically the delays. If I start making one delay different from the others, you lose oscillations, as I told you before. If you make, you need to have equal delays. So it's hard to tune the frequency of these oscillators. But in our heart, as I said, we need to go from 60 beats a minute to 180 when we're exercising. Or in neurons, you have to go from to. Or in the cell cycle, some cell cycles in the embryo are every 20 minutes. In the old person, every 2 weeks. You still need good oscillations with tunable frequency. So this is tunable. You can get, by changing a parameter, tunable frequency with the same amplitude, basically. So you can change a parameter and go from this to this to this by changing a param. So it has a kind of a different feel. Now, these are 2 extreme cases. Usually you can build circuits that go between this and this. It's not harsh categories, but this shape, when you see it, a lot of times in biology, like if you ever saw an ECG, like a heart, ding, 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 ding. That's the kind of thing I'm talking about. Okay, so that is a classic biological oscillator. Yeah.
When you have such cycles in protein translation, relative to what time scale are they narrow? Like what is the time scale with?
Great. So as you notice, you said in your question, there's a separation of time scales. There's this period here, and then there's the fast period here. And when you change the parameter we're talking about, you basically change, you lengthen this period, but the shape of it and timing of the pulses stays the same. So I want to analyze where they come from, these pulses, and you'll see that there's typically one kind of slow arrow here. So these have a separation of timescales between these 2 arms in order to get this. So they're built with a separation of timescales with positive autoregulation. And so I want to analyze one of them so we can get it. Okay, so that's the last thing we'll do. So we talked about AM, FM, repressolator, delay oscillator, this motif, and now I want to talk about just like a biological circuit, very well studied, and is a classical and important oscillator. And this is the oscillator in the cell cycle. So you have a cell, it grows, it doubles its mass, doubles its DNA, sends the chromosomes to the 2 daughter cells, splits, and you have 2 cells. Right? This takes some time. And we'll talk about, so it's actually, in fact, a very complicated circuit. It has a lot of checkpoints, so you don't divide before you've duplicated your DNA. So there's a checkpoint that makes sure you've duplicated your DNA before you can divide it. There's a lot of checkpoints like that, but there's one cell cycle in biology that's minimal. It's reduced to its minimal components because it has to work very, very fast. It doesn't wait. It waits for no one, just like time. And that's the cell cycle oscillator in the early embryo. So when you have a fertilized egg, it starts dividing 2, 4, 6, 8, 12, no, 2, 4, 8, 16, 20, 2, 4, 16, 32, 64, 128, 256, 512, 1024, and then stops. Okay, so it's like 10, 10, 7, 10, 11. Then you have a transition where it starts expressing genes. Before that, it just splits. And that's every 20 minutes. And it's really cool to see. You have an egg, you see. Amazing thing. So that oscillator is built like this, like that. So it has this X. In fact, it's a kinase that phosphorylates a machine called APC that degrades cyclin, which is a component of CDK. So we have here X and Y. And X also auto-activates itself. Now I'm just going to give you another detail here, which we'll explain later. This positive autoregulation is in fact two loops where it activates an activator and represses a repressor of itself. I'll try to explain that. And this is conserved. This is called CDC25. This is called WE1. This circuit is conserved. It's the core of cell cycle in basically all vertebrates, mammals, also in yeast. So it's like important. So it's like when you see that, you say, let's try to understand this circuit here. So this is the positive feedback is actually made of a double positive and a double negative. How does this work? So CDK works, it's a complex, it has this subunit called cyclin that makes it active. And APC degrades this cyclin. This is the way it works. So that's this arrow. So it degrades cyclin. And here phosphorylates APC. Okay. The key to understanding this, there's experiments by James Farrell at Stanford, is that what he did was, I'll tell you the answer. This depends on bistability. So to get this such decisive clock like that, you need X to have two states. If you remember the tennis ball I showed you that was cut in half and you can be either one state or the other, that's like tick-tock, tick-tock. You just need a mechanism that starts pressing on one side and it goes tick. And pressing on the other side, it goes tock, like that. So you need this bistability-based mechanism. And the way he did it, he took cyclin that can't be degraded, and he started adding it to an extract of frog eggs. So he chose the frog eggs because they're like big eggs that you can make a juice out of them and study them in the test tube, and they still show the cell cycle in the test tube without DNA and without anything. So it's like, and he measured this active CDK1, and he started adding cyclin, and it goes up, 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 up. But then at a certain point, so we have this CDK1 has a positive feedback loop. So at a certain point, it starts activating itself and jumps up. It's like the tennis ball. And then keeps going up like that. And then he did something quite amazing. Instead of adding more and more cyclin, he started here and started taking away cyclin. So if you walk up here, it goes like this. But if you walk down, when you reach this point, you don't fall because the positive feedback loop is active. And you go down, and you have to go down to a lower point here before you fall. So this bistability creates a phenomenon called hysteresis, which means that when you're going up, you have a high threshold. And when you're going down, you have a low one. So this is like, if you try to think of the shower, this situation would be that if I'm cold now, I want to go hot, I have to go way up to the hot. But once I'm hot, I have to go way down to the cold, to go to cold. This kind of switch, as you can imagine, makes it even more possible to oscillate because the information now, it's even more difficult to hit the middle because in fact you can't. And you can't avoid it basically. You oscillate like very hot, very cold, very hot. And this is a property of positive autoregulation of bistability. And so how does this work? It goes like this. So this cyclin is being made all the time. So I'm going to label this 0, 1, 2, 3, 4, because this is for this question of where these timescales come from. We go around this loop. And I'm going to plot here the dynamics. So this is CDK1 active. Now this CDK1, when it's active, this activates this proteins for cell division, right? So this is the key player here. When it's active, it makes the cell divide. And when it's inactive, the cell is not dividing. So this is like the output. So you see, it goes like this. So we start with a situation where APC is low. APC is low. It's not degrading cyclin. Cyclin is being made. It's building up, building up, building up, building up, building up, building up. When it reaches the high threshold, CDK1 shoots up to here. But then APC is made. APC is made. What does APC do? It degrades cyclin. So now we're going down. Actually, it degrades cyclin quite quickly. So we go down like this. So we go down like this. And then we fall down to a very low level of CDK, like this. So all this spike has all this spike is the explosion of CDK1 because of the positive feedback loop, the degradation of cyclin, and the drop to the low fixed point. And then the slow part is building up cyclin again. And then you get another spike. I just want to say that if you increase cyclin transcription rate, it just builds up faster. So you go to a situation where instead of this, you go to a situation of this. So that's how you can get faster spikes. It's basically, the period is controlled by this build-up stage. And this is called, in physics and biology, relaxation oscillator, because this name comes from you build up tension like that, and then you let it go. And that occurs in neurons. Like I said, the same idea basically occurs in neurons, in heart cells, with different components. Now, so bistability is okay. I want to maybe propose that at this point we do our tradition where you explain to each other this little circuit. Okay, and then we'll demystify why you need these two positive feedback loops. So turn to someone sitting next to you, please, and just explain. See if you can explain how this works. Please, even if you're hesitating, please still do it, because it's the person next to you will benefit from just having someone to listen to. אתה יכול באחוריך, גם. איך זה הולך?
צריך להגיד להם פה על המחשב, זה לא עבד.
זה היה בזבוז זמן קצת. מה זה?
לא הבנו למה CDK עולה פתאום.
כאן? פה? אני אסביר את זה. למה CDK עולה פתאום? למה זה יורד? Okay. I asked some people, and I understand that I skipped a stage in the explanations. I want to explain something. The question was, why does it shoot up like this here, right? And so the idea here is that we have this positive autoregulation, and I'll soon go into more details, but the idea with positive autoregulation is that you have this removal and production that can have 3 fixed points. And when you change the production rate of the cyclin, what you're actually doing is raising this curve, you can say, like that. And at some point, the only fixed point is this one. And then when you lower cyclin, you go back down. As you go back down, you have 2 fixed points, but since you started here, you're still here until you go so low that you have only this bottom fixed point. So by tuning cyclin, you're changing the graph from here to here and then down to here in order to make those transitions. At some point, you see this fixed point is moving, moving, moving. At some point, the graph doesn't touch the removal graph, and you jump to the high point. Now when you go down, you're still here until the graph no longer touches this part, and then you have only 1 fixed point left. So you go from 3 fixed points to 1 fixed point to 3 fixed points to 1 fixed point to 3 fixed points, but you're stuck at the point where you were just now, either high or low. So that's an important part of the explanation I forgot. Yeah.
But what sets the rate of change? From
This cyclin.
From the change, the two part, like number 2.
This part, this rate.
This rate, what sets it?
This is it's basically, I'll show you, this is phosphorylation reaction. So this positive feedback loop this needs to be phosphorylated, and phosphorylates a protein that phosphorylates it. So this is like it's like the minutes it takes to phosphorylate fully. And then when...
And this is transcription, it's not transcription.
This is phosphorylation, phospho, this here is phospho. So these are, now I get it. Now during the class I get it finally. That we have here minutes time scale, and the slow time scale here is this production of cyclin that can take a long time. So that's the, okay, other questions? Yeah.
At number 4, when it goes down, is
Yeah.
that because APC has reached some critical point to start inhibiting CDK1?
And so when APC goes high, it phosphorylates APC, and then APC starts degrading cyclin. Cyclin goes down. Cyclin goes down, and cyclin goes down, and X goes down, until cyclin goes so low that that you have to jump to the low fixed point. That's the only situation.
Biologically, what's happening in that jump down?
What's happening in that jump down is that CDK positive autoregulation no longer can support high activity of CDK, and then basically this phosphatase here just dephosphorylates it. So this is a fast dephosphorylation.
The degradation is also still a constant, right?
This
Yeah.
it's degrading cyclin, so it's not so slow.
It's not so.
No, so this is this part. Cyclin degraded, dephosphorylation. Cyclin degraded. Now let's look at this positive feedback loop here. This is totally a positive feedback loop made of 2, just to get these details. And I want to show you a cool analysis by James Farrell about so why do you need these two positive feedback loops? And the answer will be to make by stability robust.
Where do you see the positive feedback loops?
These ones, 1 and 2.
Even though that's double negative, you call it positive?
Yeah, a double negative is like a positive feedback loop, right? Why, yeah.
Yeah,
yeah.
I know.
So the way this works is this. The CDK1, I'm going to call it X from now on, is active when it's phosphorylated. So remember phosphorylation, adding a phosphoryl like that. And these two enzymes, W and Z, catalyze the forward and the backward reaction. So if there was no feedback loops at all, let's just analyze the circuit. Here is X phos. This is phospho divided by the total. So if everything is phosphorylated, it's 1. If nothing is phosphorylated, it's 0. And we have the rate of removal by Z. So the more XP there is, the more removal you can have. And we have the rate of addition, this rate of by W. So I just want to tell you that it has to go down to 0 here. This is adding phosphoryl by W. It has to go down to 0 because when XP is everything, is all X is phosphorylated, there's nothing for W to work on. Right, so these two lines, did I explain myself? Here, at this point, there's no more X0 to change to XP. And of course, they meet these two rates, adding and removing, at one point. And this is our stable fixed point. So if there was no feedback loops, this system would just reach, let's say, 50% phosphorylation. No oscillations, no bistability. No. Now let's add one feedback loop, one of the two. Let's say this one, where XP activates by phosphorylating W. It activates W. How does that change these curves? So Z is unchanged. This is removal. But look what happens to addition. When everything is phosphorylated, it has to be 0, like before. But also, when XP is low, addition is low, because it activates its own addition. So it starts low, goes high, and then goes low. Why does it start low? Because of the feedback loop. XP, you need XP in order to make itself. And you can tune it so that you have 3 fixed points here. A stable one, low, a stable one, high, and an unstable one where you go to these two others. And then you have bistability. Just like I described, that's great. What's the problem with this? We noticed this also in the lecture where we talked about bistability before, is that if I change removal rate, let's say I have more Z or less Z, I change it, I already have 1 fixed point. It's in fact very fragile to this removal rate. So if I change Z or removal, I lose the 3 fixed points, and I lose bistability. In fact, there's only a narrow range where I still get 3 fixed points. It's this range here, where it becomes tangent here and tangent here. This range here, you can only change Z by about 60% to get bistability. Do you see that? It's this getting 3 points like that requires luck, basically, too. Now let's see what happens when we have two feedback loops. This one and this one. So XP activates its own production and inhibits its own removal. This is 2 ways where XP enhances its own production, by enhancing its production and inhibiting its removal. What I like about this argument is that it's basically on symmetry. So the adding is the same as we just saw. It looks like this. And the removal is 0 when you have no XP. And it basically looks the same, except now XP inhibits its own removal. So high XP has low removal, and then it rises, and then it goes to 0. So what you have here is like two humps instead of a line and a hump, because XP increases its own production and decreases its own removal. And now this configuration makes it much easier to cross the 3 points. In fact, in order to avoid those 3 points, you have to take, let's say you need to take, which one should we take? We need to, here we changed removal, right? So this is the removal. In order to lose the 3 points, removal has to be like this, or like this. So everything in this range of morphing the removal keeps 3 crossing points. So here it's tangent to this point here, and it's tangent to this point here. So you can have a 10-fold change in Z and still have bistability. It's a kind of regulation of two opposing enzymes. And as I said, this happens from yeast to human beings. No organism we know of lost one of these. So if it wasn't needed, it would be quite easy to lose it. And this rate plot analysis shows how you can, for me, it shows how you can understand something quite elaborate just based on symmetry of these shapes. Questions? Yeah.
So can you explain that part again?
Which part?
Just what the graph is.
What this graph is? Oh, this entire graph? Or so did you? Should I start here or this one? This one? This one?
No, I understood that. I understood that part. Just you don't mind just repeating a bit of that.
What I drew here is removal and addition, and I took into account that X removal, addition. So when XP is low, addition is low, and then it rises because of the positive feedback loop. And when XP is very high, it goes to 0 because there's nothing more you can add phosphoryl to. And because of the symmetry, XP inhibits its own removal. So when XP is high, removal is low, and then XP goes down, removal goes up because it inhibits its own removal. And then at 0, there's no more XP to remove from. So that creates two humps like that. And when you have two humps, it's very easy for them to cross 3 times. Because if I change parameters, what it means is morphing, let's say, one of the humps. So I change parameters, and so now this hump is lower. It still crosses 3 points. And I make it even lower, it still crosses 3 points. To make it really tiny, in order for it to cross only at 1 point. Or to make it really huge for it to cross at 1 point. Because I make it a little smaller, it crosses at 3 points. So two humps like that are like two, geometrically just easier to cross 3 times. And in fact, 10-fold more parameter range to get bistability. And bistability is important for this tick-tock action of the relaxation oscillator. And so just to summarize, we talked about 3 different kinds of oscillations. Basically, a delay oscillator. We have like a repress oscillator. You have 3, at least a feedback node with at least 3 things. We talked about noise-induced oscillation, where you have a damped oscillator. On computer, it looks like it doesn't oscillate, but when you put it in the cell because of noise, it gets kicked, and that apparently controls transcription factors in the cells. We talked about relaxation oscillators, which have these sharp pulses that you can tune their frequency because of basically they have one slow part and then a stereotyped spike that's very fast. And that's what happens in heart cells, the cell cycles, etc. So, do you remember your part? Okay. I said, roll, baby, roll, roll, roll. I said, roll, baby, roll. I said, roll, baby, roll. I said roll. All night oscillator, oscillator, keep up your pulse with negative feedback, with delays, cooperativity with noise, with noise. I said roll, baby, roll, roll, roll. I said roll, baby, roll. I said roll, baby, roll. I said roll, yeah, all night long. Thank you for your backup singing. And there won't be a class next week, so I'll see you in 2 weeks.